This video is all about tools for anatomy, physiology, and genetics. My name is Pritina Laru. Tools for anatomy. In the late 19th century, scientists were first able to view neurons by taking a thin slice of brain tissue, applying stains or other compounds to it, and then viewing it through a microscope. Histology is the study of how cells form tissues. Histological techniques have shown the brain changes underlying some neurodegenerative disorders. An enzyme that breaks down acetylcholine is linked with plaques and tangles of Alzheimer's. With electron microscopy in the 1950s, scientists were able to tell that neurons were individual cells. Before that, they didn't know if neurons were individual cells or just a mesh of physically interconnected cell bodies. To tap the signaling pathways, radioactive molecules or tracers can be injected into the cell body of a neuron, and then the movement of the radiation can be monitored. MRI, developed in the 1980s, creates images based on the distribution of water in its tissues. MRI clearly differentiates between cerebrospinal fluid, the water-rich gray matter, which is the cell bodies, dendrites, and synapses, and the white matter, myelinated neurons. Tools for physiology. A thin glass electrode can be placed inside a neuron to measure the voltage across the cell membrane, to which changes the curve when a neuron is activated. This can be done to monitor neuron activity in living lab animals. If a slice of brain can be kept alive in the optimal environment, researchers can determine drugs' effects on the brain. These first two electrophysiological techniques are highly invasive, but in EEG, electroencephalography, 20 discs are placed on the scalp, connecting to a machine that records the neuron activity with wires. However, this is not at the individual neuron level. In two-photon microscopy, a lab animal can be genetically modified so some neurons in it produce a protein that glows when a laser is shined on it. This can be done to understand changes in the brain during normal processes like learning. Tools for genetics. To map gene locations, scientists can start by identifying a trait with a known chromosomal location that is linked with the trait of interest. CRISPR, clustered regularly interspaced short palindromic repeats, evolved as a bacterial immune system that targets viral invaders. Later, its components were used to zero in on specific DNA sequences and recreate mutations or test DNA. Optogenetics allows researchers to control brain activity with light and, for example, see how neurons work in circuits. In optogenetics, a lab animal is genetically modified so it has a light-responsive protein. Optic fibers are then inserted into the neurons so they can either activate or silence them. Genetics in neurological diseases. Mutations impacts differ between diseases and people. Lissencephaly is a disorder which has a malformation of the brain when it is smooth. It affects development. Babies with lissencephaly start having spasms in the first month of life and develop drug-resistant epilepsy and severe intellectual and motor disabilities. 70% of those with lissencephaly have LIS1 mutations, but there are other mutations associated. Kabuki syndrome is characterized by intellectual disabilities, a distinctive facial appearance, a slow growth in infancy, and other physical problems. Most have mutations in KM2D, and some have mutations in only some of their cells. There are other mutations in genes that are like KM2D. Fragile X syndrome is the most common form of congenital intellectual disability in males, and it is caused by excessive CGG repeats in the FMR1 gene. The protein coded by the FMR1 gene is important for synapse function, and it is disrupted by these repeats. Some people with excessive repeats may not be affected, but they will be carriers with the risk of passing it down to their children.